Today I will speak about the species problem and the species as the basic units for evolution and for biodiversity. Because all the people clearly know or recognize what a species is, because you have the differences, quite very strong differences between all the organisms, so easily to identify a cat or to identify a dog or a lion or a zebra and so on. This is very clear, but if the species are very closely related, then it becomes difficult to separate between them. And this is the problem for estimating evolution or for getting insights into the evolutionary processes, and especially for today to get information about the diversity, the high biodiversity. So when we look for biodiversity, we have to use an elementary object, and this is the species. Now the species in the classification process must be comparable anyway through all the species. So they have to possess some fundamental basic properties. And these basic properties must be realized for every species. So, and afterwards, when we found such properties, then we can detect or separate the species using secondary properties. And these are the so-called classificatory properties. So, for example, if you look for the citizens of Krakow, then the fundamental property are to be a citizen. And then you can classify or group them according to so-called secondary properties, the classificatory properties. So for sexes, for income, you can group them for the height, and so on. So these are the secondary properties. So what's the fundamental properties for a species? They make them all comparable. So species act with organisms. And for example, the basic property for an organism is life. All organisms have to be characterized by life. And what's life is a set also of basic properties. It's not only a single property, because it is characterized by response to stimuli, by reproduction. Life is characterized by growth. Life is characterized by development and also by maintenance of homeostasis. So in the species problem, we find now fundamental criteria that must be general in any way. If you don't find such fundamental properties, then you have the problem to mix apples with oranges. So one of the first fundamental property possess all the species they are clusters or groups of genotypes. So genotype is one of the fundamental property, or it's the first fundamental property. And now we have to ask which criteria makes the interconnectivity between these genotype pools, between these pools of genotypes, and to separate them. Then we have to find which criteria makes changes, transformations of the genotype pools possible through time. And this is called evolution, because always we found that species is the basic unit in evolution. So species have to evolve. And last but not least, we have to separate between the genopool types. And this is speciation. So we have to find such fundamental criteria. Now in the last years, not the last years, I think from the 40s on, they have developed many concepts. They are running today also. So one of the first and important concept was the so-called biological species concept, introduced mainly by Ernst Mayr. Now the definition is that species are groups of actually or potentially interbreeding natural populations which are reproductively isolated from other groups. Now, we have some disutilities in this concept because they are restricted to biparental organisms, and a species concept has to be explorative. It has to explain why there are species and how they are created, not to describing species only. So, and the biological species is explorative in some restricted way, and it is not operational. You cannot detect biological species directly by the interbreeding. You cannot look for interbreeding of all the, or test or check the interbreeding possibility between all the organisms within the species. This is impossible, so it is not operational. Now we have the next concept, and this is developed by by paleontologists, and this is the lineage-based species concept. And by definition, this concept is species are separately evolving lines, or because specifically metapopulation lines or segments of metapopulation lines. So they really describe lines, evolutionary lines, but they lack to define the causing mechanisms. What are the mechanisms to explain them? So they are not really explorative. They cannot explain how species are created, and they are describing only lines. So more or less they are operational. Then we have the phylogenetic species concept. By definition, it's very difficult because species are diagnosable clusters of individuals within which there is a parental pattern of ancestry and descendant, beyond which there is not, and which exhibits a pattern of phylogenetic ancestry and descendant among units of like kind. Now the main disutility it is a complete operational concept, completely operational, because it only explains every mutation, and later in the cladistic this can be named an apomorphic character, each mutation as an own species, the next new species. Now we have, within such biological species concepts, we have the cohesion species. So species are the most inclusive groups of organisms having the potential for genetic or demographic exchangeability. So this concept is good, but it does not explain the mechanism for the cohesions. So it is also not really descriptive for all this form. It explains in some kind, it's explorative, but it does not explain the mechanisms for the cohesion, which creates the cohesion, the interconnectivity within the gene pools. Then we have the so-called genotypic cluster concept, and this is a very good concept because species are groups of genotypic clusters that can overlap without fusing with its sibling. So each genetically different clone will be identified as a separate species, and the genotypic cluster concept describes, so it's more or less an operative concept, because it describes, it is based only on the homogeneity in the genotypes, but it does not explain why they become homogeneous. So they are not explorative but operational. Then we have the differential fitness concept. It is a very new concept defined by Hausdorff in 2011. And this explains in part that there can be some permeability between isolated genotype pools. So by definition, species are a group of individuals that are reciprocally characterized by features that would have negative fitness effects in other groups and that cannot be regularly exchanged between groups upon contact. So the disutility of this concept, this is of course not operational, but it is only restricted to selection and mutation as the main driving forces for speciation and not regarding other evolutionary factors like, for example, genetic drift and migration. Now, the last concept I have developed was the so-called, or is the so-called transferability concept, species concept. By definition, and this is a very broad and extended definition, that species are groups of organisms sharing a pool of genotypes with the potential to transfer genomes, either complete genomes or half of genomes, to the succeeding generation. And now a very important point, perpetuating transferability. And then splitting up or off the pool of genotypes without further transfer potential between such pools gives rise to new species. Of course, this concept explains now the mechanisms how a species is in which way a species is characterized, but of course it is not operational. It is a real explorative species concept. Now, from the historical point of view, but from the biological historical point of view, only species concept describing lines are useful, because such lines keeping an own evolutionary tendency, and they are distinctly separated and independent from other evolutionary lines. Now, when we regard species as such lines with their own history, then they are characterized by birth processes, because they are regarded as individuals by an individual has a birth process, which is called the speciation process. And then we have the development, the ontogeny, and this is a species transformation or anagenesis by Rentsch. The birth process is called cladogenesis. I will explain it later. And last but not the least, we have death. And death is characterized in the species by extinction or by split up speciation. Split up speciation means from a mother species it is split up in two or more daughter species. Now I will describe or concentrate on the morphological methods for delimitation of the species. And we have here 4 characters, four methods, better to explain 4 methods to find possibly morphological delimitations of the species. First, we have to regard the homogeneity in characters, character homogeneity. This should not be only morphological characters, it could be also molecular genetical characters. So it's better to describe phenetic characters. Then we have to look for the ontogenetic cohesion. So the cohesion within the development of the species. We have to look for ecological homogeneity, because a species are restricted to homogeneous niches. And we have to look for evolutionary continuity. And these are the lines. So, character homogeneity, ontogenic cohesion, and ecological homogeneity, we can regard within one time horizon. Evolutionary continuity, we have to regard between continuous time horizons. Now let's concentrate first on character homogeneity. How this can be done, the differentiation between different species or between different characters. Now there we have to look for the frequency distribution, of course. And on the above you can see there are three distinct, more or less homogeneous, characterized by normal distribution, frequency distributions. And at the base you can see such clear distributions can only be followed or detected when you have no change during ontogeny, no shape change. So from the insta1 to insta5, they keep always the same change until the adult. But this is only rarely the case in organisms. Normally, organisms change also very simply, also continuously during their life. So we have a shape change, and on the one side we can see when we have high juvenile mortality, then the summary distribution will result in right side distributions, constant mortality in platykurtic distributions, but constant mortality is very rare in organisms. And then we have low juvenile mortality, and this leads to left side skewed distributions. So normally for most organisms, and especially for invertebrates, for non-vertebrates, high juvenile mortality is the rule. So normally you will find right side skewed distributions. Low juvenile mortality you can find now, especially with humans, yes. But when you look for fossil forms, then the the larval, the small individuals, mostly are gone by such processes called taphonomic processes, and only the adult forms, or the forms close to the adults, are represented in the fossil record. And such leading to the left side distributions. Now, as an example, you can see here a foraminifer, and it is it is a single-celled organism living on the reef crest, and you can see here the change in size also as a typical character, and you can see this has a life time, a lifespan for 1.5 years, and you can see the change from smaller, of course, to larger forms, but you can see also the abundance shift. Of course, juveniles are much more abundant than the more or less adult forms. Now, when we go to the diagram with the changing ontogenies, then the separation between these three formerly clearly distinct frequency distributions are not as clear now, because you have such distributions with weak juvenile mortality, with constant mortality, and with strong juvenile mortality, and they are overlapping in some regions. But when they have more or less the same frequencies or similar frequencies, they are characterized, the overlapping region is characterized in the one form by very large individuals and in the other by very small individuals. Abundant large individuals, on the other side, a few large individuals and abundant small individuals. So, on the next slide, we have to regard ontogenetic cohesion. We have regarded first in a continuous line, but now we have to regard it in polymorphic species. So, ontogenetic cohesion can be that we have different polymorphs. And this polymorphism can be expressed in a sexual dimorphism. Of course, the sexes look different. We have many examples for this. It can be in a generation dimorphism. There are two generations. You can find them many in plants, yeah, and in many invertebrates. Yeah. So you have an asexual and a sexual reproducing generation. Then you can find social polymorphism, and last but not least, developmental polymorphism. So, an example for social polymorphism together with sexual dimorphism. Here are the termites. You have workers, you have soldiers, and you have a king, this is a male, and the queen, the female. Then we have developmental polymorphism, as we can see here. We have from the egg to the larval stadium, you have a very high changing rate. This is the embryonal development. Then we have a low changing rate, mostly growth, and this is the tadpole. And then we have the metamorphosis from the tadpole to the frog. Again, a high changing rate, but then a constant less changing rate going down to zero. A further complexity to detect relationships or connection within a species is the congruence of phenotypes of adults despite completely different ontogenesis, as you can see here in this picture. So the adult forms have the same shape, identical shape, but this shape can be gained by different ways, quite different ways in the ontogenes. So when you calculate here in the form, in a very simple calculation by lines, by a line, then you can see that the parameters of this line, of this linear equation, are quite different. So by comparing these two species, it is not good to compare the adult states because they are identical, but to compare the parameters of the linear equations. They are very different. On the one side in the multiplicative character and parameter, and otherwise in the additional parameter. Then we will go to niche homogeneity. So the response of the organisms to the environment. Here you have the illustration of a gradient, a function of the gradient, a continuous gradient, and the species react with frequency. And you have a range of good conditions, and in the middle, in the center, you have the top, the optimum conditions. Then you have a zone of physiological stress, and then you have the intolerance zone. This is a very simple theoretical scheme, but normal distributions, these clockwise distributions, are exceptions. So we can see here there are deviations from these normal distributions due to the Weber-Fechner law, that the response of the species is not, or of an individual of the species, does not follow a linear equation, but it is restricted in some kind. So as you remember, acoustic, our hearing by ears, is in various frequencies. Thus, when we look at the depth distribution of Calcarina majore, this is also a foraminifer living in deeper forms, then you can see on the left side there's the depth distribution in meters, and this is a normal distribution, can be fitted by a normal distribution. But depth is not the real environmental parameter who influences the distribution, because these forms have symbiotic algae, and so there they are Therefore, they are light dependent. But light going through the water column follows the Weber is exponential. It decreases exponential. Thus, the response of the Calcarina Majore, as you can see in the middle, is left side skewed. So it's no longer a normal distribution because Calcarina Majore follows the Weber-Fechner law, as you can see directly. To the linear scale, the response is non-linear. But when we transform the environmental condition, light, irradiation, by the logarithms, then again you will become, or you will get the normal distribution, similar to the distribution of the depth distribution. But deviations from the normal distributions can be originated by other factors, and especially by competition. Competition is one of the main factors influencing distribution or the environmental conditions. The conditions along the distribution along the gradient. First, we have to differentiate between competition by species of the same species group, of the same kinship, as it's called in biology. And as you can see to the left side, you can see the competitors only influence at the borders of the distribution. So the distribution are a little bit compressed at the borders, but also by the competitor itself. On the other side, you have competition by a different, completely different species, or even genera, but of the same group, like foraminifera. And now the range of the competitor can span throughout the whole range, spans throughout the whole range of the competitive species. So it is not restricted to the borders. And there is a mathematical calculation possible. So the frequency of the competitive form depends on of course, the original frequency and of the frequency by the competitor. And this leads to the Lotka-Volterra equation. But here it is additionally introduced a so-called fitness coefficient of species, of the one species versus the other species. Because anyway, when there are both species present, both are competitive by the other, but to different degrees. So here we can see competition examples, again, symbiont-bearing foraminifera, single-celled organisms, living almost deeper than these shallow living forms we have shown before. So these are two groups. On the left side, you can see different frequencies, but the one with the green color is much more compressed. The distribution is much more compressed, much more influenced by the other form, because the fitness coefficient is much lower, additionally much lower than the other form of the, in this case, Operculina complanata and Operculina elegans. To the right side, we have two forms of the species group, and they have the same fitness. So a little bit more competitive influence in this case by the so-called Planistigina longicepta is only caused by the higher frequency. So they have no competitive influence of competitive fitness. Now an example from the influence, competition between different species and different genera. Here now we have the influence of a competitor from a quite different species and also a quite different genus, but still belonging to the symbiont-bearing foraminifera. The distribution in this case is not a normal distribution from the suppressed Heterostegina depressa. It is a right side skewed distribution, but in this fact you have on one transect you will find a bimodal distribution, and on the right side you will find directly a normal but left skewed distribution. Now the question is, which causes, or the question was, what are the causes why this, in the first transect we find a bimodal distribution, and we are looking for a competitor, and in this case it is Calcarina majore, and you see with a much higher fitness coefficient in comparison to the Heterostegina depressa on the right side with 0.28 against 0.72. Now on the left side in that transect, it's so-called northern transect, Heterostegina is suppressed by Calcarina majore, because in that transect Calcarina majore is very abundant, and in the other transect it is, you can find only very few specimens. Such the distribution comes close to the normal frequency distribution of Heterostegina depressa.